The next part of medical biochemistry is to think about now what our food breaks down into. So we looked at the process for food traveling through our digestive system, but now what's actually happening on a molecular level to our foods. So I want you to take a few seconds here, think about what your favorite foods are. What types of molecules are then in those foods? So if you're not really sure, I guess I would try to first guess what it is. Let's say you like pizza. What parts of pizza are there? What is pizza made up of? What toppings do you have? What things are in that pizza? But then what are the molecules that make up those components? For example, cheese. What's in cheese? And if you're not sure, maybe try to first guess. And then if you're still not sure, then you can kind of go online, look up some nutrition facts. It'll let you know what types of things are in cheese. So you can see what kinds of fats or carbohydrates or proteins are in your different foods that you're interested in. We're going to start looking at mostly what we're looking at for what we can consume and what we can take in to generate energy for our bodies are going to be carbohydrates that are going to be made up of starches and different types of sugars such as sucrose and lactose, and different types of fats including triacylglycerols, and also dietary proteins, which will break down into amino acids. When we're looking at the different types of carbohydrates, again, I mentioned there are starches and different types of sugars such as sucrose, lactose, glucose, and it all comes down to glucose is the fundamental smallest component of the polymers of carbohydrates, but it's all about how they're bonded together. So different types of bonds between those glucose molecules are going to dictate if it's a starch, if it's sucrose, if it's lactose, cellulose, glycogen, for example. So you can see down here, starch, these long chains of glucose molecules, and that's shown in these forms here, right? The hemiacetal form of glucose, but then there's going to be a reaction that's going to be bonding these together. So in starch, it's one bond in one way. In cellulose, you can see those bonds are flipped, so every glucose is kind of upside down compared to the other one. And then glycogen, there's long chains, but then there are side chains that come off of that. So this is all going to be dictated, what they are is actually going to be dictated by how they're bonded to each other. How they're bonded to each other, for example, we can name these different types of bonds. For example, alpha 1,4, and the alpha is if it's, they're straight, they kind of are on the same directionality, or if they're flipped. And then the 1,4 bond is referring to the number of the carbon in the chain. So 1, 2, 3, 4. So it's the 1 and 4 carbon that are bonded in an alpha orientation that creates this kind of bond here. Why the orientation is important is because different enzymes recognize different shapes. And these different shapes are going to look different if they're bonded upside down, or they're bonded in 1,4 or 1,5 or 1,3. If they're bonded at different regions, they're going to have a different structure. And that means that a different enzyme, such as sucrase, right, is going to recognize sucrose, this compound that has a glucose and a fructose component. When the sucrose is bound by sucrase, it hydrolyzes this bond and breaks it down to glucose and fructose. Another example, lactose is recognized, it's got a beta 1,4, so that's the opposite of this kind of bond here, right? Lactose and lactase hydrolyzes this bond, so it breaks that bond, and then we generate galactose and glucose. So lactose is a common element in our dietary carbohydrate in milk that can be broken down into galactose and glucose. You may have heard that it is important to get fiber, increase your fiber. And the reason fiber is good, and we're going to talk about this in a little more when we go back to the digestive tract, but fiber is good because our body, we don't actually have human enzymes that can break down fiber. This link for cellulose, for example, a component that's often found in plant tissue, it's got a beta 1,4 link. Now, we don't our body does not recognize that link. So we don't break down cellulose. So what it actually does is it's kind of the dietary fiber is helpful for our bodies because it actually helps push things through our digestive system. It helps clear things out, and that is why fiber is good for us. So this is as opposed to a starch, where starches we can consume those, where starch is a repeating subunit of glycogen that has an alpha 1,4 link instead of a beta 1,4 link. So this small change in, it's the same monomer, but it's just organized in a different way. We can't recognize that, and it completely changes our ability to digest that and consume that for energy purposes, but it still has beneficial health benefits. The other thing we can do is we can mess around with the structure. So you'll see on the left is sucrose. It's one of those sweet types of foods that you can eat if it has sucrose in it. Our taste buds recognize that, and we enjoy those sugary flavors. The artificial sweetener sucralose is recognized by our tongue, but it's not recognized by some of the enzymes in our body, in our human digestive system. So you can see here, difference from sucrose and sucralose is you'll notice instead of these hydroxide components that are bonded, they're actually chlorine molecules. So you can see these hydroxide are changed to chlorine molecules. There are some other minor changes here. Over here, we've flipped this bond. But what we're able to do is we're still able to get the sweetness factor from sucralose, so it tastes sweet, but our bodies generally do not recognize this bond or these other components, so that enzymes can't bind because these chlorine molecule or atoms are actually interfering with the proper binding of our enzymes, so they pass through our bodies undigested, and then we don't consume those as calories then. So there's a lot of worry about artificial sweeteners and how they may be harming you. I just want you to be careful about that. I mean, I think everything is you should kind of think about doing in moderation for artificial sweeteners or regular sugar for that matter. Obviously, we know that it has negative health benefits. But be careful of some of the sources you're looking at. So I found this source on Mercola.com, the dangers of Splenda and sucralose. So I was kind of doing a little bit of research just on the internet. I want you to take a few seconds to read this, and then I want you to decide based on their description, and then I want you to look at the molecules down below of DTT, sucrose, and sucralose. You decide. Does this make sense? And kind of write down some of the things that you think, for or against this. And again, look very closely at the molecules and note some of the differences between them. And I'll be interested. We'll probably have a conversation about this in discussion, but it's just kind of an interesting thing. I'd like you to kind of take some time to look at this. And we talked about the digestive system already, but I just want to talk about some of the enzymes, some specific enzymes that recognize and digest dietary carbohydrates. So maltase is one of those. It's going to maltase, isomaltase, lactase, sucrase, and alpha dextrinase. So these are all going to generally, right, maltose is the sugar that they're going to recognize, or lactose, or sucrose, or dextrose, sorry. So these are all, you notice that they are named after the sugar that they are able to interact with. And the ASE ending, this ASE ending here, that's going to dictate that it is an enzyme, right? So make sure that you recognize that ASE ending is suggestive that that component is an enzyme. I also wanted to point out where some of these enzymes are released. We do actually release some enzymes in our saliva. So we have a salivary amylase that starts to break down. So it's a minor breakdown, but there is some breakdowns of starches and other sugars in your mouth. Have you ever noticed if you've had certain types of gum that you've chewed for a long time, you start noticing they kind of start falling apart? It's because some of your amylase activity is able to break those starches or some of those cellulose components apart a little bit, or those sugary components apart a bit, and it starts to make the gum kind of fall apart as well. So you have some enzymes that have capacity to break some of those things down. There is then, once it gets down into your stomach, the salivary amylase, the pH changes, and that influences the enzyme activity. We're going to talk more about enzymes later, but there's not much enzyme activity that's happening in the stomach. It's more of a pH change, a denaturation, and a breaking apart of some of that the food that you're eating. We then have the release of pancreatic enzymes, such as another amylase from the pancreas, the cytokeratin peptide hormone also, so CCK is another enzyme that's released to help break down proteins and amino acids. And then in our enterocytes, in our small intestines, we have these disaccharides are cleaved into monosaccharides. So a lot of these different enzymes, maltase, isomaltase, lactase, sucrase, dextranase, those are going to recognize smaller chunks of carbohydrates and then break them down into monomers and dimers that can then be recognized by the brush border enterocytes and then be brought into the cell. So here's an actual electron micrograph picture of a brush border of one of those enterocyte cells. Here is an image of one of those, just a cartoon image of an enterocyte, and here's the basal end and the apical end with these little fingers that you can see here. This is where we have all of these transporters, and sometimes they're sodium glucose transporters, they're co-transporters, glucose or galactose or fructose. So they generally have different transporters that recognize those different amino acids. Now again, sucralose doesn't have a transporter. It is not recognized. Those chlorine atoms on that molecule do not allow it to get into these transporters. So they get rejected, and then they travel through the enterocytes through the small intestine without getting taken up by the cells. So once they're taken into the cell, they can be manipulated to some extent, but they're generally just processed through the glucose, galactose, and fructose, gets shuttled through the GLU transporter, and it is sent over into the basolateral membrane, into the blood system. So one of the things that cells do generally, once they get glucose or any of these other components, they'll turn those into glucose. We'll talk about the metabolic processes for that. But the transporters, actually, we can go both ways. So we can release glucose back into the small intestine, or they can come into the cell. But what usually cells do is they'll trap those glucose molecules in the cell by phosphorylating them as soon as they come through. So we immediately, as the glucose comes in, it becomes phosphorylated, so it becomes glucose 6-phosphate, and that's actually the first step of the glycolysis cycle and many pathways that glucose is used inside the cell. And what it does is it allows it to trap that glucose inside the cell, because then it can't pass the membrane. They're not able to, the transporter no longer recognizes it, and so it gets stuck inside. So we can increase the amount of glucose in the cell, even if there is low amounts here, because we trap it in once it gets passed through. So our blood glucose levels, then, once that glucose gets into our blood system, that's going to be regulated by various hormones that we'll discuss in a second here. Really low levels of glucose is hypoglycemia, high levels hyperglycemia. Obviously, low blood glucose levels can cause weakness, rapid heartbeat, and can cause mental confusion. Also, I like to say it can cause some hanger issues. So you might not feel that great. You might be ornery a little bit. Obviously, prolonged hypoglycemia can cause major issues and permanent dementia, potentially, if your body cannot regulate its glucose levels. Hyperglycemia is also not good, right? We want to be in the normal range between 60 and 110. 110 is a little high, even pre-diabetic, really. But when you have higher than normal blood glucose level, that can cause increased urine flow and osmolarity balances in your blood. Your blood gets kind of thicker, and your kidneys have problems processing the blood as well. And it can cause low blood pressure, coma, death if it lasts too long. It can cause tissue damage because of that osmolarity. So the osmolarity is just the amount of things that are dissolved in your blood. And if that there's too much sugar in there, that can cause problems. Your cells don't know how to respond to it. And that's why a lot of times people with diabetes sometimes have to have amputation of their limbs, their fingers or their toes, because the blood's not flowing properly there. The way that the sugar is regulated in our body is through two different hormones that we're going to talk about. One is insulin, and it's released when blood glucose levels are high. So when we have pancreatic beta cells release insulin into our bodies, and what that does is that helps to open up, that brings in glucose, enters the cells faster, so our cells will express more glucose transporters, so we can bring more of those into our cells. It helps to increase glycolysis, and we haven't talked about glycolysis, but I'm sure you've seen it before in other classes. So that's the breakdown process of glucose. It increases glycogen synthesis in the liver and in skeletal muscle. So glycogen is a long chain, it's a polymer of sugar, of glucose. And so that's kind of the way that we store quick energy in glycogen. So it starts to build up those stores in our muscles and in our liver to use later on when they need it. And it also activates the synthesis of fats, so lipids. So insulin increases fat, glycogen stores, and starts to break down some of that excess energy to create energy. The other hormone is glucagon, and the easy way to remember this is glucagon is turned on when glucose is gone. So it's released in the blood by the alpha cells of the pancreas. And so then basically the opposite stuff happens as when insulin's there. Glucose entry into the cell slows down, glycogen breakdown in the liver speeds up. So those are the storage of sugar polymers, right? Glycogen is broken down and released into the blood, so we can increase that blood supply of glucose. Lipids, so fats, start to break down when glucagon is turned on into raw materials to synthesize glucose. So our liver is able to make glucose from fat, as well as make fat out of glucose. And gluconeogenesis accelerates. So that's the generation of glucose from these fat components. The hormones glucagon, released from the pancreatic alpha cells, and insulin, released from the pancreatic beta cells, control abnormal blood glucose levels. Hypoglycemia and hyperglycemia. Define hyperglycemia and the hormone that controls it. So you should be able to answer this question, and if you cannot, then you should rewind this and try to figure that out. And now I'm going to be talking about some of the things that happen as someone is going through a fasting state. So this chart on the right is looking at the relative change of different factors. So insulin and glucagon, those hormones are in dotted lines. Some of the different macromolecules like glucose in blue here in the solid line, the fatty acids with the brown line, ketone bodies in the red line that I mentioned a few lectures ago, and liver glycogen as well. So blood glucose versus liver glycogen. And what you'll notice that there are metabolic changes that occur in the absence of food, with a gradual decline in the blood glucose concentration. And this is accompanied by an increased release of glucose from the glycogen stores. So the glycogen stores begin to go down, but then that's because this glycogen is being broken down, and then that allows the blood glucose levels to remain relatively stable instead of traveling down this line just like the glycogen does. So initially there's a breakdown, and then it stabilizes because the glycogen is broken down. So all cells contain glycogen, but most of it's stored in the liver, about 90 grams in a 90 kilogram person. And it's also in muscle cells, so about 350 grams in a 70 kilogram person. So muscle and the liver stores much of this. So the muscles obviously have a need for quick energy, and so a lot of it's stored in our muscles as well. So the idea with carb loading is to kind of build up those glycogen stores for people that are kind of going through doing some athletic competitions, for example. Free glucose and glycogen represent less than 1% of the energy reserves that are used, and they're usually used up in about 15 to 20 hours of normal activity. So that's normally just like a normal day. If you're going to run a marathon, they might be gone in about 3 hours, however. Should also mention protein. It's used up at a rate as high as 75 grams per day, but we're able to recycle that. So we don't need to take in all of that. We don't need to take 75 grams of protein a day because we're able to recycle a lot of anything that we break down or use. We also, I should mention, lipid catabolism is also mobilized after fats. As I was talking about before, when we have glucagon in the system, we break down those fats. And when we have a lot of those fatty acids being broken down, we create a lot of acetyl-CoA, and that's metabolized into ketone bodies that are used in our bodies, in our blood, in our brain for energy. And so this happens after a few hours. We increase the amount of ketone bodies so that our bodies can use those for energy as well. And we'll talk about that more when we get into some of the details of metabolism, the mechanisms there. So what do you think here? When blood glucose levels rise following a meal, which of the following events do you think occurs first? Take a second, pause, and then resume when you're ready to go. So we should expect that insulin levels should rise because your blood glucose levels rise, right? So the answer there is number 4. Now we're going to go from lipids to fats, so dietary lipids. I mentioned this before, right? We have an ester group here, and on that ester group we have this kind of base component. If we had hydrolyzed this and make this an OH and OH and OH, this is glycerol. That's the main kind of backbone component is glycerol. And then these are fatty acid tails. So we have that carboxylic acid functional group. You can kind of see if this were to leave this group, obviously it's an ester now as it's attached to this carbon, but you can see that could be a carboxylic acid functional group. That's why I say 3 carbon chains esterified to glycerol. This is the glycerol, and this is an ester functional group. Most of our chains on the back end here, those tails, are 16, 18, or 20 carbons. They're usually even numbers, and that's just how we manufacture fats in our bodies, so they're kind of even numbered groupings. You can see here this is a saturated fat, and that means that they're not shown obviously here, but each of these carbons has 4 bonds, and then they're saturated with hydrogen. In the middle here, this is a mono unsaturated fat, so we have a double bond, and then a polyunsaturated fat here. These are all cis fats, right? Because these bonds are kind of bending down in the same direction. There's also omega fatty acids. There's a numbering system on the back end, and you can count back, and then the omega fatty acids, those are the fatty acids people tell you want to consume. That's all defined by where that unsaturated spot is from on the back end, and it's just defined by a numeric system there. So if you ever see omega fatty acids, it just means that there's a unsaturated component on the omega carbon. Cholesterol is also considered a dietary lipid. We modify cholesterol for steroids and hormonal components in our bodies. It also incorporates into membranes and changes the fluidity of membranes. And we're also going to talk about it as a cholate precursor. So we use this to kind of help flush our bodies of other components, and I'll talk about that when we're making bile salts and such. So I will discuss that in just a moment here. The dietary lipids also can be converted. So once we eat these, we can convert these into phospholipids. You'll notice this phosphate group here is a change compared to what, this ester bond that we see otherwise. We can modify one of these chains, and we can create different types of components for cell membranes. So you'll notice that we have this polar head group. So that's why when you see things like this in biology, this is what we're actually talking about right here, that polar head group. And then these longer tails are shown here. In addition to some of the fats, there's also, I wanted to introduce you to the fat-soluble vitamins as well. These are the dietary vitamins, the fat-soluble vitamins. I like to think of them, if you have to ever remember them, AID, K, ADK, or ADE and K. And so these are important vitamins that we use within our bodies for different functions. For example, vitamin A helps to convert light into neural signals. And so retinoic acid is also a signaling component that is involved in vitamin A signaling. Thymine, riboflavin, niacin, NADH, FADH2, they're involved in the production of in the electron transport chain, essentially, for generation of energy. Carotenoids are another group of pigment molecules that are found in various plants that help give pigmentation for different colors of vegetables. If you want to look at different peppers, for example, right? There's green and red and yellow and orange, and the carotenoid, the small changes will change the color of light they absorb and change their pigmentation. But they're also really good antioxidants. These are really good at taking up energy or taking up free radicals and kind of sequestering them, taking them out of our bodies. So that's why people say to eat a lot of vegetables and get those antioxidants. They help to absorb some of those potentially mischievous compounds in our bodies. And I mentioned I was going to discuss with you some of the importance of our cholesterol. In our stomach, some of the stuff that the muscles in our stomach are doing is they're undergoing this peristaltic churning. There's also the release of bile salts and acids and cholic acid and deoxycholic acid. So primary bile salts are actually modified cholesterol components. And so what happens, you can see that there's actually two sides. They're ambipathic, so they have a kind of a polar side and a non-polar side. So you can see here, this is a cholesterol-like compound, and we have these hydroxide compounds that are going to be able to hydrogen bond, and this carboxylic acid end, but there's still a lot of hydrocarbon here on this backbone of the cholesterol. So the polar bonds lie on one side and the other side can interact with the lipids, so then it can kind of break down fats into these smaller bits and interact with them and help to break them apart so that we don't have these big droplets, because our enterocytes cannot take up giant droplets of cells. They have to be broken down into smaller components and subunits. These also, cholesterol or these bile salts, also can interact with our dietary fiber. So it also helps take some of the fat and some of the other compounds out of our body when we want to get rid of some of our cholesterol. If we have a lot of fiber, it can pull some of these bile salts out of our body along the track with our fiber. So that's all how we can also get rid of some of our cholesterol, and why it's thought that good amounts of fiber can also reduce cholesterol levels. You can kind of see here are some examples of modified bile salts, cholesterol. They're, again, ambipathic, and glycocholic acid, we have modification of that. Carboxylic acid, we talked about, it's a secondary amine, right? And then this is an amide formed that's added on. And then here's an amide formed as well. This is taurocholic acid. We have a sulfur group on here, but this is kind of the backbone of cholesterol, and it changes slightly because of to modify it for different bile salts. And lipids are generally, when they're broken down into these smaller teeny bits here, they're stabilized by in these mixed micelles. So we have cholesterol, we have bile salts in here, we have monoacylglycerols, lysophospholipids, free fatty acids. So we have lipid hydrolysis. I mentioned earlier on, when we can, in a different lecture, right? We can hydrolyze this bond and break off this into a free fatty acid. So some of these free fatty acids are in here when we add lipases, colipases, different types of compounds that are broken apart. So some of these are secreted as zymogens. So this zymogen term, that refers to something that's released as a precursor. And that precursor, when it interacts with the low pH of our stomach, it actually turns into the actual enzyme. So it gets activated in the gut of our stomach. So why do you think that those compounds would be released as zymogens? Why wouldn't they just be released as active components that are active enzymes from our cells? So I would like you to think about what do lipases do? Where would they find lipases? Where are lipases found? And then why do you think lipases, how do you think they're activated in the stomach? This is a diagram of kind of what is going to happen to those components, and this is just a different picture, right? So it's a box diagram instead of a chylomicron in here, or a mixed missile. But they have their free fatty acids, monoacyl glycerol, for example, vitamins, the fat soluble vitamins, cholesterol, phosphatidylcholine, for example, different bile salts. They're in that missile, and then they get taken up by the enterocytes. So this other box is another cell, the enterocytes. So this is the apical side of that with those fingers that would take up these components, and they're able to then get taken up by the cells. They're processed within these enterocytes. So some of the cholesterol and phosphatidylcholine are repackaged. Proteins are synthesized, ApoB488, ApoA1, and they're put on and they're tagged onto these new chylomicrons, so these little droplets of fat, and then those are released into the lymph system and into our bloodstream. And then we have phosphatidylcholine, we have different fats, the vitamins that our cells need, and these can travel through our blood and be taken up by other cells. So when people look at your lipid concentrations in your body, they're looking at some of their high-density lipoproteins or low-density lipoproteins, right? HDL versus LDL. And some of the differences there are the high-density lipoproteins. They're usually going to be transported. These tags are going to dictate if they're high density or low density. And what we're looking for is the HDL is generally going to be tagged from tissue that's coming from the liver. So high density lipoprotein is telling the system that it's actually removing cholesterol from the system and converting these into bile salts. But when we're going for low density lipoproteins or very low lipodensity proteins, that's actually endogenous fats and cholesterol. It's transporting them from the liver to the other tissues. These HDL is going to the liver, and LDL, the liver is sending out. And usually what that means is that if we have high levels of HDL, we're cleaning up our body. If our liver is sending out these fats, that means that we have a lot of fatty liver, and it needs to do something with them. So we're going to shoot them out into our bloodstream. So generally, when your doctors look at your fats, your cholesterol levels, they're looking at differences between HDL and LDL. They used to just look at the total lipid concentrations and be like, well, you have it's not good. You have total lipid concentrations are too high. But now they're actually looking at ratios of HDL versus LDL. And it kind of tells us something about our system and how it's processing fats, and if we're cleaning up our system or if we have too much and our liver is sending fat out to deal with it in, for example, to make more adipose tissue or go into other regions. So now I just wanted to briefly touch on this idea of enzymes and how they function and work a little more active in a little more detail. So this is just showing you on the left here is an enzyme in this darker brown shade. This is an enzyme, and that's the active site. And so structure is really important, right? And where the amino acid side chains are located in that enzyme are really important. And the substrate, the substrate is also important for what it looks like and how it's going to interact. For example, we're looking at proteins now. We're going to switch from fats to proteins. In the stomach and the pancreas usually secretes some different enzymes that help to break down protein. So the stomach is more about denaturing that protein, so breaking down the quaternary, tertiary structures and maybe the secondary structures. And now the primary structure is broken down by these enzymes. they're hydrolyzed proteins, so we break down that amide bond. And it's usually we recognize different side chains, different proteases are going to recognize different side chain functional groups and bind them, and then we're going to be able to enzymatically and quickly break apart that amide bond. So again, about 70 grams a day dietary protein are processed, and then 60 to 70 or 90 are recycled in our bodies. So we don't constantly need to consume all of the protein we use. We do recycle a lot of it. So this is important. Again, we have a lot of zymogens that are released so that in our bodies as peptidases, and some examples of some of the enzymes that cut proteins are trypsin, chymotrypsin, proelastase, so different types of proteins that are going to have different recognition sequences that they bind on amino acids. So the same thing, once those larger proteins are digested into smaller amino acid chunks, then there are receptors that can recognize either single amino acids or smaller chain peptides. Larger peptides are not taken into the cell, and they're kind of recycled back, and they need to find some enzyme that can break them down before they can be brought in. So there are some peptidases in the system on the brush border that actually recognize those, and they'll break them down right on the surface of the cell. And then we have amino acid transporters. They're brought in, they're metabolized or changed within maybe some of these inside our cell. We have some enzymes inside the cell that can break down into smaller amino acids, and then those are released into the blood system. Trypsin prefers to cut downstream the C-terminal end of positively charged residues. So I want you to look at this and predict the products if trypsin were incubated with the peptide shown below. So pause and unpause when you want to look at this again. I'm going to just walk through this one here just to make sure that everyone's on the same page for what I want you to know about amino acids and peptides. So one thing is, the first part is downstream the C-terminal end. This is the N-terminal end, C-terminal end. You'll notice the N-terminal ends and amine group, and the C-terminal ends a carboxylic acid functional group. Trypsin is going to cut downstream the C-terminal end of a positively charged residues. So these, the side chains, also referred to sometimes as residues, and we have to figure out which one's going to be positively charged. Well, normally under normal physiological conditions, we don't have charges on this cysteine or the serine molecules. You can look that kind of information up on any of those charts of amino acids. But physiologically, on the end here, this is kind of tricky. It's a trick question a little bit. Usually under physiological conditions, the amine groups are going to be positively charged. So at a pH of 3, usually you're going to have a positive charge on the end here. Also probably have a positive charge here and possibly here as well. So it is a little tricky because I didn't put that on here because I wanted you to think about that a little bit and try to look and use some outside sources for answering this question. Because you can, because you're at home and it doesn't have to be off the top of your head, but I wanted you to look that information up. So if I'm looking at downstream the C-terminal end, well, downstream, if I'm going to cut here, right, well, we already cut downstream here. Downstream is generally traveling from left to right, and by convention usually we have the N to C. Just like in DNA, we always go 5 to 3 prime. For amino acids, we always go N to C. 5 to 3, N to C. So downstream here of the C-terminal end, we're going to cut. Where can we cut? Well, remember we make this is a carboxylic acid derivative, right? So we're going to cut this. We're going to form, to make a new amino acid, we're going to form, oops, sorry about that. That's not what I wanted to do. I just want to make a carboxylic acid, right? We're going to form a new carboxylic acid here, and then we're going to have an amine functional group here. So we'll have a new N-terminal and a C-terminal end. We'll have a new amino acid. So this is how those peptides are going to break down. So we're going to just cut off this lysine group, and then this other cysteine, serine, arginine peptide is going to remain intact if we add trypsin to this peptide. So these are some of the glucogenic amino acids. So they're some of the amino acids we can bring this into. Once they're broken down into, for example, this lysine, we can actually convert these into, or we can shuttle these into the glycolysis cycle or gluconeogenesis cycle. So for example, we can alanine, cysteine, glycine, threonine, tryptophan can all be made into pyruvate, and pyruvate's an important component, as we'll see in a little bit, of that glycolysis cycle. Alpha-ketoglutarate, succinyl-CoA, fumarate, oxaloacetate. So a lot of amino acids will be able to be entry points into the metabolic processes we'll talk about later that generally involve glucose or glycolysis. Sometimes there are some enzymes necessary for converting these amino acids into these steps, but we generally have those available to us in our bodies. And finally, the big thing, once that we do take these amino acids and convert them into the citric acid cycle or glycolysis or gluconeogenesis, we have to deal with the nitrogen, because that's the one thing, that's the one unique thing about amino acids, is there is a nitrogen on that compound. So when we have nitrogen, we have to have a way of getting rid of it and removing it here. So these are some examples of the nitrogen cycle in our bodies. And so we can use this. We have to take that nitrogen, and generally we take this out of our bodies to create urea. So this is a way that we can get rid of nitrogen in our bodies in a safe way, and we can essentially urinate this out, and we can get rid of excess nitrogen in our bodies in this way. Especially when we're using some amino acids for energy purposes to break off that nitrogen into, and then use the rest of the carbon chain of the amino acid for energy. And that is the end of the metabolic biochemistry portion.
Next, we're going to focus on how our bodies utilize these macromolecules that I've shown you how they break down to create biochemical or cellular energy.